Welcome to the Diabetes Bio Podcast, a podcast for the Diabetes Journal, the flagship research publication of the American Diabetes Association. Each month, we will interview one or more authors from the feature articles, the papers of the month, or other articles or events associated with the Diabetes Journal or the American Diabetes Association, respectively. We also have a Sweet Talk segment of the podcast, which is more of an open format discussion of additional articles, conferences, or major events in the field that we'd like to highlight. My name is Darleen Sandoval, and I'm joined by my podcast co-hosts, Dr. Kirk Habiger and Dr. Kevin Williams. Happy Tuesday. How are you guys doing today?
Hey, Dar, doing good? Happy Tuesday.
Good, doing well.
All right. So each interview, we ask our authors a surprise icebreaker question that they are not made aware of before the interview. So in honor of our Women's Interprofessional Network of the ADA interview, which Kirk is going to tell us about in a little bit, I'm going to put both of you on the spot and ask you to name an influential woman that you admire, either science or non-science. Kirk, I'll start with you.
I actually have to thank this woman in science. I was a lowly technician out of undergrad, and Dr. Lucinda Carr at Indiana University School of Medicine, she really pushed me like you would a student, and even to the point of sending me back to classes, which eventually led me to pursue my PhD and I guess stuck me here. So Dr. Carr is my influential woman who happens to be in science.
Oh, that's great. Kevin, do you got one?
Yeah, and we didn't pre-plan this, so we're sticking with, I guess, Indiana University School of Medicine, but I was really impressed with Carmela Evans Molina, which we interviewed last month, and I was really taken aback by how inspirational she is and how she approaches her life of wanting to learn things new every day, not just science, but outside of science. She had many inspirational phrases for the younger generation for scientific training. And I've already used some of those phrases in a lecture I gave just about a week ago,
Nice.
highlighting her and her work. I really appreciate her coming on and giving that type of an interview.
Yeah, I don't know Dr. Carr, but I do know Dr. Molina and have known and been aware of her work for a long time. And she really is, she's great. She's admirable. So mine is actually someone that I've been interacting with quite a bit the last couple of years. And I had the opportunity to interview her last year for ADA, and that is Barbara Corky. She has a very interesting story. So she, especially considering when she developed her academic career. So she actually worked as a research technician before she went to graduate school. And the fact that she, persevered and became one of the leaders of our field, I think is just really inspirational. Plus, she's just a lot of fun. All right. So, Kirk, why don't you tell us what's on deck for today's episode?
Sure. Thanks, Dar. This month, we will talk to the authors of the paper of the month from Dr. Dax Fu and his group at Johns Hopkins. This paper details how masking of the antigenic exposure of beta cells via a monoclonal antibody against zinc transporter 8 suppresses the immunological cascade from beta cell antigen presentation to T cell mediated beta cell destruction, providing a novel islet-targeted and antigen-specific immunotherapy to prevent and reverse clinical type 1 diabetes. We're also going to hear from the co-chairs of the Women's Interprofessional Network of the ADA, WIN-ADA, as Dar mentioned. Those investigators are Drs. Jennifer Lee and Rebecca Scalzo. They're going to tell us more about WIN-ADA and what events they're hosting during this year's ADA scientific sessions.
All right. And Kevin, what do you have in store for us for the Sweet Talk segment?
Yeah, today we're going to talk with Dr. Olga Gupta and Dr. Rana Gupta from Duke University. They both contributed an article to the Classics in Diabetes series examining previously published works in the journal that discuss the impact of regional adiposity or depot-specific fat distribution on metabolic health. I think many of us are familiar with the apple versus pear body shape analogy when discussing metabolic health. Olga and Rana really offer some valuable insights that go beyond this analogy, exploring the heterogeneity and quality of adipose tissue in relation to metabolic health. So stick around to find out more.
That sounds great. Thanks to both of you. And it looks like we have an exciting episode. So let's jump right in.
Today, we're going to discuss the paper of the month from the May 2024 issue of Diabetes, titled Cell Surface Zinc Transporter 8 Antibody Prevents and Reverses Autoimmune Diabetes in Mice. We're joined by the lead authors, Dr. Devi Kasinathan and Dr. Zheng Guo, and by senior author, Dr. Dax Fu. Dr. Fu is an associate professor at Johns Hopkins School of Medicine, and Drs. Kasinathan and Guo are postdoctoral fellows in the Fu group. Welcome, Devi, Zheng, and Dax. Congratulations on your paper, and thanks so much for being a part of this month's podcast.
Thank you.
All right, we like to start with something a little bit different to get the conversation rolling. So I'm going to ask you an icebreaker question. And the question is, tell us a fun fact about yourself that has nothing to do with science. And we'll start with Dax.
My fun part, actually, I used to ski and rock climbing a lot when I was young, but now my kids grew up, so I cannot do all the rock climbing anymore, but I actually is transporters to carry all the pet and the equipment for him and to the rock climbing around the world.
Nice.
Very cool.
Yeah.
Debbie?
Fun part, I'm not enjoying anything. I always come to lab and do my work and happy. I play with my mice and then I back and then I just sleeping, eating nicely. That's it.
It's just what Dr. Fu wants to hear, probably.
That's right.
Zheng?
Oh, actually, I'm very happy to join this lab, and Devi and Dax is my best friends in my life and my academic life. So I'm very happy to work with Devi and Dax. Yeah.
Okay, great.
So we're delighted to have the lead authors of this paper with us today. However, Dr. Fu, we'd like to give you the opportunity to highlight any other contributing authors or collaborators to this work.
Okay, so we are new to the diabetes research, and also we're new to the animal studies. So this work is not possible without the support, guidance, and expertise from our collaborators. Type 1 Diabetes Project started with collaborations with Dr. Lee Yu at the Barbara Davis Center for Diabetes, University of Colorado. He introduced us to the autoantibody, the clinical immunology. And Lee also introduced the Aaron Michael, see the therapeutic potentials in our project and provide the T-cell immunology expertise and advice on the potential the clinical translations. The Will Wong and Maria Golson are our Johns Hopkins colleague at the Baltimore campus. A part of the mice, the studies was done in Wong's lab with the help of his student, Darleen Saba. And the histological experiment was performed in Maria's lab under her personal watch. And the human islet experiments are essential to validating the mice result. And we are fortunate to have our Johns Hopkins colleague at the Florida campus, Matt Poyle and his associate Chandon Sona. They both did the MEV43 uptake the experiment during the Christmas time. The Maria at Baltimore campus provided us the human islet to perform the cell surface binding experiment in our lab. So indeed, the MEV43 project is really bringing us together on a very enjoyable, fun experience. And also we have the very productive the collaborations in the past 2 years. So there are many interesting questions remain to be answered. So we work on the diabetes communities to use the MEV43 as an islet targeting reagent to advance the diabetic research.
Yeah, great description of team science there. Dr. Fu, can you now go ahead and briefly summarize your research program and maybe a bit about what led to this specific project?
Okay. Basically, our research program has been focusing on the zinc transport exclusively for over 20 years now. So we begin with studying the zinc transport structures by the X-ray crystallography. And in 2007, we determined the structures of a ZNT8 ortholog from a bacteria. These structures show a unique Y-shaped dynamic structures, like the immunoglobulin IgG. So we anticipate the antibody binding to the ZNT8 can cross-link ZNT8 to produce some amazing biological consequence, but we know nothing about that. So then in the same years, the ZNT8 was first time linked to the type 2 diabetic by GWAS. and also identified as a common autoantigens in type 1 diabetic. And moreover, ZNT8 was found exclusively expressed in the pancreas, the islet, and responsible for the zinc enrichment in the insulin secretory granules. So however, the ZNT8 the deletion in mice models did not affect insulin secretions and changes the glycemia controls. This is quite intriguing to us. So we pivoted to found the structural biology to ZNT8 cell biologies. And we found that ZNT8 is a dynamic protein is distributed between the endoplasmic reticulum and the insulin secret granules. And also ZNT8 in the ER is highly sensitive to the pro-inflammatory cytokines. And the glucose stimulations can induce a transient cell surface display of ZNT8 on the beta cells, but the surface of ZNT8 can rapidly internalize when the cell will switch to the low glucose. The finding of the cell surface presentation of the ZNT8 antigens, this prompted us to focus on the cell surface ZNT8, the autoantibody. So now, after identifying the cell surface MAP40Z3s, now we have an opportunities to work with the expertise in the diabetes field to explore the potential these therapeutic mechanisms in mice models. And in the long term, we hope to translate it to the human use.
All right, Devi, I'm going to follow that up with you and ask, as Dr. Fu mentioned, you guys are focusing on ZNT8 in this particular study as an autoantigen in patients with new onset type 1 diabetes. But maybe you can expand a little bit on what you think this protein is doing or autoantigen is doing in normal physiology, but then what you hypothesized was happening with type 1 diabetes.
So I joined the lab in 2021, bring my surgical skills for islets isolations and animal care. So the zinc transporter 8 is generally considered a zinc sequestrating transporter in the insulin secretory granules. So, however, our lab found that it is a dynamic proteins in equilibrium among the ER and insulin secretory granules and cell surface membrane. In healthy settings, the zinc transporter 8 is responsible for zinc enrichment in the insulin secretory granules. So the zinc is required for insulin packing in the beta cells before glucose stimulated secretions. In type 1 diabetes, zinc transporter 8 is a major autoantigens recognized by both autoreactive T and B cells, contributing to autoimmune regulations and progression in type 1 diabetes.
Great. Now, Zheng, the M antibody 43 is designed to prevent immunological detection of zinc transporter 8, correct?
Yeah, it's correct.
Are the other antigens present in the beta cells more vulnerable to immune attacks when you do antibody 43 treatment?
Yes, actually, our current mechanism of action involves masking cell surface ZNT8 from the immunological action. And we recognize that ZNT8 is just one of many autoantigens involved in type 1 diabetes. Autoimmune progression is radio process consisting of the sequence of epitope spreading events. So different autoantigens contribute to disease initiation and progression differently. My earlier autoantibody study with Dr. Yu suggested that ZNT8 may be an initial autoantigen in type 1 diabetes. We do not know if MAP43 and ZNT8 binding can block epitope spreading. If it does, MAP43 may prevent autoimmunity progression to recognize secondary autoantigens. But actually, this is one of the many working hypotheses to explain the protective effect of MAP43 in NOD mice. We are also exploring other alternative mechanisms, actually. Yeah.
Thank you.
Devi, back to you. This antibody had the unexpected action of stimulating B cells to a Treg fate. Is this exclusive to the islet, or is this a systemic response of this MAB43 treatment?
So the zinc transporter 8 is exclusively expressed in the pancreatic islets in the human body. Since zinc transporter 8 is not expressed in human cells and MAB43 does not have the cross reactivity to immune cells, the in vivo MAB43 effects on Treg cells stimulations is indeed an unexpected findings. Since MAB43 does not directly modulate the immune cells and its immune regulatory effect is secondary to a direct MAB43 effects on beta cells. We are now investigating the beta cells response to MAB43 modulation in NOD mice to understanding how a direct beta cells modulating by MAB43 propagate to increasing the Tregs in the islets.
Great.
All right. So, Dr. Zheng, we just heard that zinc transporter 8 is specific to the beta cell. Aside from that, is there something specific about protecting that transporter, or is this strategy amenable to maybe a cocktail approach where multiple targets are protected that will give additive or even synergistic overall protection against type 1 diabetes?
Yes, actually, as all we know, among the four major autoantigens in type 1 diabetes, VNTH actually is the only one surface autoantigen identified so far. And IL-2 and GAD65 are considered intracellular antigens. At least there is no solid biology evidence to their cell surface display in a high density like VNTH. And the cocktail approach, I think it could be a great strategy to achieve additive or even synergistic protection. But however, there are no alternative cell surface antigens that are well established to be targeted by antibody masking. So regarding the difference between the VNTH and other autoantigen, it may be worth mentioning that the ER-resident VNTH is highly responsive to pro-inflammatory cytokine stimulation. In contrast, IL-2 and GAD65 are not a response. As pointed out by Devi earlier, the protective effect of MAB43 may be beta cell centric. We are now investigating beta cell response to MAB43 modulation in NOD mice to understand how directed beta cell modulation by MAB43 induced broad protection against the polyclonal T cell attack, not just the VNTH specific T cells.
Wonderful.
We'll finish off with you, Dax. Can you highlight any of the strengths, but maybe also the limitations that you see with this study and how you would like to address that in future directions?
Sure. Yes, I just want to echo the point we have already made today. And basically, the VNTH does not directly modulate the immune cells. Its action is localized in the pancreatic islet, but the safety profiles of MAB43 in NOD mice suggest that the VNTH can be used for the long-term suppressions of the type 1 diabetes progressions. And the VNTH can also use to reverse established type 1 diabetes in NOD mice. So although the beta cell proliferations was observed in the islet of the NOD mice experience this remission, we did not observe the beta cell expansion at a meaningful rate for the clinical benefit. So since the insulin is a multigenic hormones for beta cell growth, and insulin is also used after avoid diabetic onset in the patient care. So we are just wondering, whether combining MAB43 and insulins can induce the beta cell restorations in diabetic NOD mice. And if the answer is positive, then what is the translational potentials to treat a patient with new onset type 1 diabetes? And these important questions is how the masking a single VNTH autoantigens can lead to the suppression of the T cell response to non-VNTH autoantigens. We observe that the MAB43 reverse the insulin autoantibodies in NOD mice. So the innovations provide the indirect evidence for the suppression of insulin-specific autoimmune response. So now we also know that the VNTH can effectively prevent the type 1 diabetic inductions by adaptive transfer of polyclonal T cells from diabetic NOD mice. So this again, they lead to the intriguing questions. How a CNT at specific modulations of beta cell can lead to the blockade of polyspecific T cell response. So we really appreciate the opportunity provided by the diabetic to raise these important questions. And also we are now working on to find answers to that.
Great. Thank you so much.
Well, just to close, Devi, Zheng, and Dax, thanks so much for joining us. Thanks for sharing your expertise with us today and talking about your recent work on masking the antigenic exposure of zinc transporter 8 in beta cells to treat type 1 diabetes. I encourage our listeners to read the full article, as our papers of the month are freely accessible online and are in the May issue of Diabetes. So this month, we're going to take the opportunity to highlight one of ADA's membership engagement groups, the Women's Interprofessional Network of the ADA, or WIN-ADA. Today, we have with us the co-chairs of this year's WIN-ADA Advisory Board, Dr. Jennifer Lee, Scientist 2 at the USDA Human Nutrition Research Center on Aging and Assistant Professor at Tufts University School of Medicine, and Dr. Rebecca Scalzo, Assistant Professor at University of Colorado Anschutz Medical Campus. Welcome to the podcast, Rebecca and Jennifer.
Thank you so much for having us.
Yes, thank you.
So, Darleen, you were also instrumental in the start of WIN-ADA. Can you tell us about how this group got started?
Yeah, so it was a little bit of luck and good timing, I think. So we started in 2017, and what happened was I had been participating in an organization group at University of Michigan with Durga Singer and Bridget Gregg, where we organized this women's career development panel, and it was really successful. It was sort of a local university panel. And we decided we were really excited about how enthusiastic everybody was about it. And so we decided to look at opportunities at the society level for more engagement and networking for women. And we realized that ADA didn't really have anything like that at the time. And so also as a part of that sort of luck and timing, I was on the organizing committee for the scientific sessions at the time. And so at the next organizing meeting, I proposed that we put together a symposium or something like that to engage our women membership. I got introduced to board members. It so happened that Jane Roos was president of the ADA at that time. And it was proposed to her. She loved the idea. And when Jane loves an idea, she runs with it. And so that was basically how it formed. And so, just a reminder of why this sort of engagement is necessary. 50% or more of graduate students and medical students nationwide are women, and yet only a third of our full professors are women. And so we have this sort of leaky pipeline, it's called, where women just aren't making it to the higher echelons of academic status. And so the mission of WIN-ADA is it's a members only network for female clinicians, scientists, educators, and other health professionals in diabetes. And the group's goals are to strengthen the voice and presence of women in diabetes research and clinical practice, to recognize the significant contributions of women to the diabetes field, to provide career development opportunities, and to increase research efforts on women's health and sex as a biological variable in diabetes. So it's really about engaging the women members of ADA and ensuring that their amazing work does not go unrecognized. So I'm really grateful to ADA and to the staff of ADA for supporting this organization. Jennifer Maisel was instrumental initially, and now Nikki Sasong, they've been really keeping us on track. We have an amazing advisory board, and we've had a lot of succession of chairs, including Rebecca and Jen, that are continuing the efforts since we started in 2017. So I'm really grateful to have the organization and grateful to have Rebecca and Jennifer continuing our efforts.
Awesome. And that was a great background of the program. You know, Rebecca and Jennifer, we always start with an icebreaker question, to just kind of ease into this conversation. So this is the same question for each of you. Can you give me your favorite role model in women's leadership? And that can be science or non-science, and I don't care who goes first.
Oh, I'll go. My mom. So my mom is a dietitian by training and was a faculty member at a community college teaching dietetics and kind of found herself, she was department chair and then working her way up. She went back to school and got her PhD in higher education, community college leadership, and was then college president for years and still to this day consults in trying to identify ways to strengthen leadership in the community college space, but still with her heart in dietetics and nutrition. And she's just been a great role model of someone to look up for that, we can do it. Women can do everything.
Very cool.
That's awesome.
Jen, how about you?
That's such a hard thing to follow up on, Rebecca, to say your mom. I'll be perceived as an ungrateful daughter now. I would say more broadly, I've been inspired by Michelle Obama and how her transition from being a lawyer to a professional figure, first lady, and now as she transitions on past that role, she's still continuing to advocate for women and girls as they develop in all aspects of their careers across many spaces. And also, I really respected her focus when she was First Lady on introducing or incorporating movement and staying healthy and active for the nation. So her memoir and her books have been really inspiring when I do get the opportunity to read non-scientific literature.
That was really great. All right. So what inspired you guys to get involved with WIN88? Let's start with Jennifer.
I'm currently an assistant professor. So earlier on, a few years ago, I really wanted to find my people within my field. And I very much strongly aligned with the WIN88's mission of increasing the visibility of women and recognizing their significant contributions to the diabetes field. And my mentor is a leader in the field, Barbara Kahn, and I went to ADA for many years when I was in her lab. And when I found the WIN88, it was an opportunity for me to expand my network within the diabetes research field. And it continues to be a fantastic community of like-minded professionals within which I've made new peer, colleague, and mentor connections. So I'm very humbled and honored to serve as co-chair with Rebecca and continue its mission through our many career development and research opportunities.
Awesome. Rebecca?
So my postdoc mentor is Dr. Jane Roosh. So I don't think I had a choice in joining WIN88 when it started, but I am so grateful that I did and have continued to obviously grow within this organization. I particularly think the career development opportunities are fantastic and a big bonus for why people should join, just through things like our longitudinal course, which I'm sure we'll talk about today. We have a lot of opportunities to connect and grow and develop. I also like the focus on women's health research. My research interest lies within women's health and diabetes, so I get a lot of support for the work that I do outside of WIN88 through this organization as well.
All right, Rebecca, we're going to come back to you again. WIN88 gives out an annual award at the scientific sessions in honor of Lois Yovanovitch. Can you tell us who Lois was and what inspired an award to be named after her?
Yeah, so Dr. Yovanovitch was a prolific scientist in the field of diabetes and women's health. She herself lived with type 1 diabetes, and that drove her research that was focused on neonatal outcomes for women affected with type 1 diabetes and really spanned her entire career. She was an icon in the field of women's health and diabetes research.
Very cool. And I guess as a follow-up, Jennifer, can you tell us who this year's winner is and why that individual was selected?
Yes, so this year's Lois Yovanovitch Transformative Woman in Diabetes Award recipient is Dr. Francine Kaufman. Dr. Kaufman's career achievements are impressive in both breadth and scope. She is trained as a pediatric endocrinologist and internationally recognized for her pioneering work in developing, improving, and testing new therapies and technologies to treat and prevent types 1 and 2 diabetes in youth. She has served multiple leadership positions, including being a past president of the ADA, as well as being a delegate of the 55th World Health Organization Assembly. And academically, Dr. Kaufman has published nearly 300 journal articles, 50 book chapters, and has led multi-center NIH-funded trials to develop diabetes treatment options for youth. She is truly a one-woman crusade fighting diabetes and obesity from bench to bedside and an inspiration to all in the field. The WIN ADA is delighted to name her as this year's award recipient.
She's also been really supportive of WIN ADA, so it's really great to be able to honor her with this award this year. Jennifer, every year WIN ADA organizes a professional development seminar at scientific sessions. Can you tell us what this year's topic is and who the presenters are?
Absolutely. We're really excited about this year's topic, which is how to say no so you can say yes, optimizing time management. Presenting on the topic will be Dr. Linda DiMeglio, who is a pediatric endocrinologist and professor of pediatrics at Indiana University School of Medicine. She is a leader in the field of pediatric healthcare with a focus on type 1 diabetes prevention, beta cell preservation, and developing new technologies and therapeutics for youth. She's also vice chair of TrialNet. She is a dynamic clinician scientist and speaker, and we are very much looking forward to her presentation this year. We hope to see many ADA members at the seminar, which will take place on Saturday, June 22nd from 11:30 to 12:30.
Awesome. Linda's a great speaker. That's going to be a good session.
Definitely.
So, Rebecca, you touched on this or mentioned this earlier, but one recent activity that is likely to have a lasting impact is WIN ADA's longitudinal course. Can you tell us more about what that is?
Yeah, I'd love to. The Longitudinal Course is an interactive program that provides professional development opportunities for early and mid-career ADA members. The course includes 3 online sessions that cover a wide range of topics that include leadership skills, self-advocacy, networking, professional communication skills, and work-life balance. We end this course with a final session in person at scientific sessions. And this course is supported by funding from the Leona M. and Harry B. Helmsley Charitable Trust. What's really great about this financial support that we get is all of our participants receive financial support to attend the in-person session and also scientific sessions for ADA. Also important with our course is we provide mentoring for all of our participants. We match participants with a mentor in their field, and participants meet with these mentors after each session to review the presented materials and determine how to best apply these lessons to their personalized professional development.
Very, very good. As a follow-up, remind us when these applications are due for next year's cohort.
We will start advertising for the next cohort during the scientific sessions. So you should be able to see advertisement for it there in person, but also in your inbox. And then we will open applications at that time.
Awesome. The next question is for Jennifer. So this wouldn't be a networking organization without some networking time. So can you tell us a little bit about that networking reception that happens at ADA scientific sessions?
Yes, absolutely. So the WIN ADA networking reception is organized by members of the WIN ADA, but it is open to all ADA members. It's a fantastic opportunity to see and reconnect with old colleagues and friends in person with the WIN ADA membership and ADA network at large. And it's also a great time to meet new WIN ADA and non-WIN ADA members and expand one's network within the academic diabetes community. And during that reception, we will present the Lois Jovanovic Award and recognize abstract winners from 4 research areas, which include sex as a biological variable and women's health, basic science, the third being behavioral science at medicine, nutrition, education, and exercise, and the last being the clinical diabetes epidemiology and diabetes complications area. The abstract submission deadline is April 25th, so if it's not too late, and this podcast is published in time, please submit your work to be considered for an award. This year's reception will take place on Friday, June 21st from 6:30 to 8:30 p.m. Light hors d'oeuvres and refreshments will be provided, and we hope to see you all there.
All right. So another question for both of you. Are there any other activities that you would like to highlight or future activities that WIN ADA will be leading that we should be aware of?
So the WIN ADA will be contributing to the ADA's Spotlight the Science series, where we interview female clinicians, scientists, educators, and other health professionals whose career contributions have had broad impact in their respective areas of diabetes work. Selected speakers have also helped to pave the way for the next generation of younger female professionals in the field of diabetes. We recently interviewed Dr. Barbara Corky and have a great list of women queued up for this year, so please stay tuned.
Great. And Rebecca, anything else to add?
I'd just like to highlight the abstract awards, like Jennifer's already mentioned. They're a great way to get your science in front of this group of individuals because you're presented with the award at the networking reception. So everyone hears your name and sees your face. It's a great way to interact with WinADA.
Very good.
Rebecca, how can people get involved if they're interested?
So if you don't have a mentor that forces you to check the box, like I did, you can, when you register, complete your membership renewal every year for ADA, you are given the opportunity to just check a box. I want to join WinADA. And there's a little blurb about it there. If you've already renewed your membership for the year, you can just go into your member profile and the box should be there available for you to check. If you're already a member of WinADA and you want to become more involved, we solicit applications for our advisory committee every early spring. So you should get emails about the application opening up, and we really invite everyone who wants to be involved to apply to be part of our advisory committee.
Awesome. She forced you with love though, right, Rebecca?
Everything she does is with love.
Awesome. All right. Well, thanks so much to Jennifer and Rebecca for sharing the work you're doing with WinADA. I really appreciate it as a woman in science. We'll put the link to the WinADA website in the show notes, and I encourage any listeners that will be attending scientific sessions to attend both the seminar and the networking reception in support of WinADA. Thanks so much.
Thank you for having us.
Thank you.
Welcome to another edition of Sweet Talk. I'm Kevin Williams. We're joined today by Drs. Olga Gupta and Dr. Rana Gupta. Dr. Olga Gupta is an associate professor and a physician scientist in the Department of Pediatrics at Duke University School of Medicine. She is a specialist in pediatric endocrinology, diabetes, and metabolism, and board certified in obesity medicine. Dr. Rana Gupta is the W. David and Sarah W. Stedman Distinguished Professor in the Duke Molecular Physiology Institute. He has a research interest in understanding various aspects of adipose tissue biology, including transcriptional programs and molecular mechanisms of how adipocytes are formed and maintained throughout the body. They are both here to discuss the Classics in Diabetes article that they wrote for this edition of the journal. It focuses on the role of regional adiposity as a factor in the development of diabetes. Thank you both for joining us today.
Thanks, Kevin. I appreciate the opportunity to be here.
Thanks, Kevin. This is great. We're excited to join in.
We usually like to start with an icebreaker question. Outside of research, what is one hobby or passion you have that may surprise people? Let's start with Olga, and then we can move on to Rana.
So I actually enjoy hip hop dance class. And there's a
Awesome.
dance studio here in the Durham area. And I think that hip hop's a great cardio workout. You get to listen to fun music and learn some moves to use at the club. We actually have performances with the costume and all of the things that
Nice.
come with it.
Do you have a special signature dance move?
You'll have to come to the show and find out.
All right. Rana?
I mean, my go-to is like so many other sports. I'm a basketball junkie. I just love basketball. Can't get enough of it. And I like to play golf. I like to play golf as much as possible. I wouldn't call myself a golfer, but I go out there and play golf. And if you could call it that when I play, but yeah, enjoy it. Enjoy just, everything sports related and then, enjoying those activities with my kids.
Are there any faculty games going on at Duke? Any walk-on activities?
Well, this is a big basketball town, so
Definitely.
I dare not challenge anybody in basketball, but certainly there's no shortage of basketball fans around here. But it gets pretty contentious, out in the community, UNC versus Duke. So
Definitely. That's great. So your article revisits some really seminal work that linked regional adiposity, in particular abdominal or visceral fat, with an increased risk of diabetes. Maybe you could start off by outlining what regional adiposity is and why or how these studies and others have suggested it as a significant concern for metabolic health.
When we talk about regional adiposity, I think what we're talking about is the accumulation or expansion of adipose tissue in certain anatomical regions. And we all know that we have fat cells and adipocytes throughout the body, but these fat cells are invariably organized into sort of distinct, what we call depots or tissues, and they're in different parts of the body. And for all intents and purposes, we think of them as sort of different tissues altogether. And there's been this link for many years that we've all talked about how body fat distribution is really a strong predictor of your susceptibility to metabolic disease and diabetes and insulin resistance and cardiovascular dysfunction. We thought it would be nice to kind of go back and revisit some of that literature. And some of it was published in Diabetes, the journal Diabetes, and heavily cited. And the one that we chose to highlight was one that we thought was really one of the first ones to, studies that actually represents a prospective study, a study that really highlighted how the accumulation of intra-abdominal adipose tissue can be predictive in a sense that it can kind of predicts whether you will go on to develop insulin resistance. And this was a study published by Ohlson and his colleagues in 1985.
That's great. Olga, that there is this concept of healthy versus unhealthy adipose tissue, or those that are overweight but metabolically healthy. Can you describe maybe what differentiates that's healthy versus unhealthy adipose tissue, or the metabolically healthy versus metabolically unhealthy overweight individuals? What is the maybe cause of driving those changes in overall health?
Absolutely. I know that we always think of weight, like the absolute number, as being linked to health, but it's really location, where is that weight stored? And the metabolically healthy individuals tend to have more of that pear shape where the weight is stored in the subcutaneous tissue. And that appears to be a protective depot compared to the more apple shaped individuals where the weight is stored centrally or in the visceral tissue. And those individuals are prone to developing the metabolic complications such as diabetes, high cholesterol, liver disease.
Yeah, and Kevin, what's so one of the reasons why we wanted to sort of bring up this problem again in talking about this, what we think is a classic article, is that since the study in 1985 and many other studies that were linking, as Olga pointed out, this sort of apple shaped, or others called it this male shaped or androgen shaped obesity. More sort of refined studies using DEXA scanning and CT scanning and imaging techniques were trying to understand, what is it about the intra-abdominal adipose depots? And it's not so clear that it's really the visceral fat. And that's one of the things that we talk about here. Is the visceral fat really the culprit? Or is it the lack of healthy subcutaneous fat? And a lot of the initial studies were kind of measuring waist to hip ratio. Of course, when you measure waist to hip ratio, you are not actually measuring abdominal subcutaneous versus intra-abdominal fat, including visceral fat and like omental fat and retroperitoneal, for example. And a lot of the follow-up studies that we talk about in this article really highlight the fact that it may not be that visceral fat per se is the culprit, but it's the lack of healthy subcutaneous fat. And there's a number of studies. I will refer to some studies from Sam Klein and Mike Jensen that really highlight the fact that perhaps sub-Q is a better predictor. But with respect to visceral fat, Sam Klein has shown that the amount of visceral fat really is associated with insulin resistance only when there's also ectopic lipid accumulation in the liver. And so there's a lot of things that to suggest that it may not be simply mass and location per se. And one of the things that we think now in recent years is that maybe it's more about the quality of the adipose tissue. And we've seen examples of mouse models from Phil Scherer and others, and the original GLUT4 transgenic made by Barbara Kahn many years ago highlighted the fact that you can have, in principle, these animals that have extreme obesity but are metabolically healthy. And if you look at their adipose tissue, it is sort of expanded in a healthy way. And so we think that, maybe the quality of the adipose tissue, which is determined by how it expands, could be an important determinant of whether or not metabolic dysfunction will occur in obesity.
I'm curious too, classically, definitions of metabolic health have been defined by body mass index or BMI. And there's been, from these studies and others, a suggestion of waist to hip ratio that should be considered. And now you are discussing points of waist to hip ratio might not fully account for the depot-specific adiposity that could be contributing to deficits in metabolic health. Is there a push to try and incorporate measures of these depot-specific adiposity for assessing the metabolic health of individuals?
Yeah, great question. I think it's a question I get a lot from clinicians and those who are studying, doing sort of translational work. What are markers of healthy adipose tissue? There's not that many that are good specific markers, but one thing that does come to mind, of course, is adiponectin. Adiponectin is a great marker of insulin sensitivity and being an adipokine, a good marker of adipose tissue health. So that may be one, but certainly we don't have good sort of markers of that. But now, of course, you can do imaging approaches to assess it, but that's not going to be clinically accessible all the time, right? Or at least not routinely. So that's still, it remains an important question of, and I guess Olga can speak to this too, what is the best way to identify those who are at risk, right? And it's particularly concerning, given the fact that we have a younger population, right? Our kids, the adolescents and kids that are developing obesity now. And I'll let Olga speak to that.
Yeah, I agree with you, Kevin. I feel like the BMI misses the mark for so many people. There are people who are at risk for complications where they're not able to generate healthy subcutaneous adipose tissue, and so they're not on the radar for potential complications. Or there are people who are, targeted excessively because of a high BMI, and they're actually not developing these complications. So the BMI really does miss the mark for a wide variety of patients. In terms of clinic tools that we have available, I think the next best step is the body fat analyzer, which a lot of the obesity clinics use, including our clinic at Duke. And I feel like that kind of helps distinguish, you're continuing to gain weight, but that weight's muscle versus fat. And so that's actually a good thing. So that's a step in, I think, the right direction, but there's definitely room to go in terms of identifying which patients are at risk. And Rana brings up a good point that the pediatric population continues to grow, and the prevalence of obesity continues to rise in the pediatric population, particularly the younger kids, even ages 2 to 4 is continuing to rise. And so this is an issue that we need to pay close attention to.
With advancements in technology such as single cell and spatial transcriptomics, are there any new insights that we're gaining about adipose tissue function, depot-specific function, how that relates to metabolic health?
Yeah, I think this is a really exciting area. And we in recent years we've now seen from the field a collective effort to derive what you might call a cellular atlas of adipose tissue, and human adipose tissue, as well as some in rodents. And I think what we've learned and what we've kind of suspected, but now we have a really good sort of resolution on this issue of heterogeneity. That the adipose tissue is remarkably heterogeneous. We've known that, but now we have an idea as to how heterogeneous it is in adults with obesity. And now this technology is going to allow us to look at this heterogeneity in different sort of disease states under different metabolic conditions. And I think that's the next step. But I think what we're learning is that when we talk about adipose tissue heterogeneity, we're not just talking about these different depots, but we're talking about at a specific tissue level or depot level, the types of adipocytes, the types of immune cells, the types of sort of vascular and stromal cells. They're quite heterogeneous, and there's functional implications for all of that. And now the challenge is to figure out what could be sort of causative, if any, in determining the health of the adipose tissue, and then how that may change with age or with treatment, et cetera.
And I think we have time for one last question. I was curious, where do you see this field of adipose research heading in the next decade? What are some key questions that still need to be answered?
Yeah, I think I can chime in with my thoughts on the basic science side, and maybe Olga will have more to say about where we need to go in terms of helping those with obesity. But we think a lot about adipose tissue and this sort of body fat distribution problem as it relates to obesity and the development of diabetes and metabolic disease. But one of the things that we try to talk about a little bit in this commentary in diabetes is the fact that this problem of body fat distribution is really, goes beyond the issue of obesity and diabetes. And, for example, in other disease settings, I'll just throw out a few examples. Crohn's disease, you have the creeping fat, right, of the mesenteric fat is expanding, and we don't understand why and how. And then, related to cardiovascular disease, you have certain depots, the periaortic fat depots that are expanding, and that could be becoming also sort of contributing to inflammation that and can contribute to various forms of heart disease. You have adipocytes accumulating in bone marrow, which can affect bone health. So the list goes on, and I think that the issues of where fat accumulates in the body, where fat cells, I should say, accumulate in the body, is going to have implications for lots of different diseases, and including cancer. So I think it's something that continues to be an important area and an issue that's unresolved.
And from a clinical perspective, two thoughts come to mind. The first one is to really, I think we need to be more inclusive of our definitions, because the cutoffs for BMI really don't take into account like the South Asian populations and other populations of people who, are having a tough time getting access to medications, who need them, but because they don't have the strict cutoff for weight or for the BMI, they don't qualify for them. And so I feel like our definitions need to be adjusted or even changed. I see us incorporating other factors like satiety and hunger into the conversation, because that's really how these drugs are acting. They're acting on the level of, of the brain and those hormones. And I think that the conversation will shift to, what's the crosstalk between those hormones and adipose and, just a whole picture view of the body.
Perfect. Thank you both, Dr. Olga Gupta and Dr. Rana Gupta, for joining us today. Be sure to check out their Classic in Diabetes article in the May issue of the journal titled The Expanding Problem of Regional Adiposity. To our listeners, thank you for tuning in to this edition of Sweet Talk. Please follow, like, and subscribe for more Diabetes Bio content.
Thanks to all of you for joining us on the Diabetes Bio podcast. We hope you're enjoying our discussion of the work in the Diabetes Journal and what is published each month. The feature articles we discuss in each episode are freely accessible at diabetesjournals.org/diabetes and are also listed in the episode notes for your reference. As with any podcast, it really helps us if you like, rate, follow, and subscribe. And if you have any feedback for us, we'd love to hear that as well. We look forward to presenting to you next month.